Hello, and welcome to the Drug Discovery World podcast. My name is Giles, and I'm here to take you through another topical and insightful article from Drug Discovery World. Today's episode is taken from a recent article in our winter 2016-17 issue, titled A Strategic Vision for 21st Century Drug Discovery. We've split the article into two episodes, and this is part one. The article was written by Dr Wayne Bowen, Dr Giles Sanders, Dr Andreas Werdich, Dr Brian Warrington, and Dr Elizabeth Farrant. With so many contributors to this article, I'll be talking through their backgrounds at the end of part 2, once you've heard all the content. So now on to A Strategic Vision for 21st Century Drug Discovery, part 1. Delivering genuine therapeutic innovation is more challenging and complex than ever. Despite major scientific breakthroughs and technological advances leading to a greater understanding of the etiology of diseases, it is generally accepted that the current approach to creating new small molecule therapeutics remains fundamentally inefficient. Bringing a novel therapeutic to the market is a lengthy process requiring endurance and commitment. Scientific and technological advances and greater understanding of the underlying mechanisms of disease are needed to fuel the discovery of new treatments and cures, but the overall cost of research and development are being driven higher by greater regulatory stringency and longer times to market impact discovery funding. Ideally, the drug-like properties promised by a discovered molecule will be safely delivered as the expected therapeutic response in a human patient during the development phase. How much time and cost should be expended on ensuring that the promise is actually fulfilled requires a careful balance of expediency and effectiveness, and there is nothing more wasteful than late-stage failure in the clinic due to impaired efficacy or safety. Being effective is far more important than being expedient. Effectiveness depends not only on generating and demonstrating a potentially safe, effective, and deliverable ligand to the target in model experiments, but also on the validity of the target as an essential mediator of the target disease. As a viable drug is the only certain validator of a target and vice versa, and target validity is unalterable, for as long as this paradox remains unresolved, the best achievable project security is that of striving to identify a superlative rather than an adequate ligand. We present an analysis of past and current drug discovery approaches, exposing their relative strengths and weaknesses. From this, a new strategic vision is suggested that can contribute to a resilient, sustainable biopharmaceutical industry to satisfy the demands of 21st century healthcare. After nearly 25 years of being a passive partner in the passive process of screening large collections of known compounds, in the hope that happenstance will provide a desperately needed cure for a significant unmet medical need, the vision is one in which the active application of accumulated and newly emerging knowledge will force the direction of drug discovery towards these goals. The concept of exploratory and directed drug discovery is not new. Quite the contrary. But during the fallow years of its use in large pharmaceutical companies, many new and relevant technologies have become available. Their incorporation into the exploratory drug discovery paradigm has the potential to be a disruptive tool for the discovery of novel and exclusive drugs. A brief history of drug discovery. The time before high throughput. Man has always engaged in drug discovery. In early times, the shaman sought out exogenous materials with recreational, curative, restorative, disinhibiting, or poisonous properties. The knowledge extended to observations that certain natural products were best gathered during a particular season, that some could be purified by sublimation, and that active agents could be co-administered to synergize or mutually annihilate their pharmacological effects. Clearly, the shaman understood the role iterative closed-loop hypothesis-led experimentation, think and try, as a means of extending knowledge to make real and novel progress in science and technology. Only the nomenclature has changed. The shaman's basic mantra, get, test, think, try again, being rephrased in terms of reflecting the technology, in this case, drug discovery. Here we see it as a succession of hypotheses that can be tested experimentally by the production of an appropriate molecule, each new hypothesis being based on an intelligent assessment of the several biological test results obtained in the previous cycle. In every cycle, the method will either find a solution better than that already known or fail to do so. Either result has the value in extending knowledge of the structural and electronic factors governing activity for all of the properties assayed. The process ends when a developable molecule is obtained or when consistent failure can no longer be tolerated. The cyclic process can be seeded in a variety of ways: screening hits or leads, natural molecules, active molecules marred by unwanted properties, active molecules with no known mechanism of action, or even molecules designed de novo with a theoretical chance of activity. The process is most effective when several assays that can measure key drug-like properties, for example potency, selectivity, bioavailability, and toxicity, are used in parallel so that the different structure-activity requirements for each of these properties can be considered jointly in the analysis and design steps to target molecules whose holistic property profile will be most likely to succeed in development. The 1980s and the shift in approach. A threat perceived by large pharma during the 1980s was that genes identified by shotgun sequencing and reassembly of express sequence tags, ESTs, could be patented by third-party sequencers, and this could prohibit them from being therapeutic targets except by paying a large license fee. The success of the Moore's Law style increases in productivity in genomics, arising from automated high-throughput sequencing and analysis of large sample and data pools, and that this could result in the complete disclosure of an entire human genome, also excited a view that the application of similar high-throughput automation could energize other components of the drug discovery process, particularly chemistry. From this developed the notion that if a sufficiently large collection of drug-like compounds could be assembled and screened, the drug discovery process could be turned into a singular, linear, automated workflow notionally suitable for finding leads for all possible therapeutic targets. The foundation of this process was to be pre-validation of gene products of the human genome, believed to be therapeutic mediators. Each was to be subjected in turn to high-throughput screening, HTS, to find drug leads in an existing compound collection. To improve the chance of success, the collection would be augmented in size and diversity using automated high-throughput chemistry, HTC. The hits found by HTS would then be verified and qualified by filtering through a succession of assays in a so-called hit-to-lead, H2L, process to find any compounds meeting the more advanced biological and chemical criteria for a lead. The lead could then seed a more intelligent process to explore congeners within the lead chemotype, to find a molecule with the more optimal properties needed to meet the preclinical criteria of a development candidate. The effects of the new approach. To those designing the new approach, compared to the automated ELAN and high-throughput paradigms of genomics and screening, the iterative cycling of medicinal chemistry to identify a single highly customized drug candidate for only one particular therapeutic target was seen as inconvenient. It stood in the way of a more expedient lead discovery process that could serve all targets proteins generated from the human genome. On balance, it was decided that the opportunistic gains to be garnered by a more expeditious path of identifying novel therapeutic proteins and automatically matching them with a pre-prepared lead contained in a huge library of candidate leads outweighed the compromises made in terms of lost rigor, scope, novelty, and would probably output a different candidate for development. The key limitations of the linear process were: first, the axiom that a therapeutic target is only validated by a viable drug is replaced by a concept of pre-validation of a target based on relevance to the disease rather than essentiality. Second, chemical scope is limited. For practical reasons, such as maintaining target throughput and library management and curation, the size of the compound library that can be physically displayed for screening, of the order of 106 molecules, amounts to only an infinitesimal representation of the drug-like universe as defined by Lipinski's rule of 5, so there is no possibility of achieving comprehensive display drug-like chemical entities. Rumsfeld's razor further restricts the choice of molecules to those known or designed to be active at historical targets and so cannot contain the unknown compound needed for an unknown target. These problems can only be solved by either an exhaustive lead library for HTS, a physical impossibility, or an active iterative closed-loop search supported by appropriate target-based or phenomenological, phenotypic assays and intelligent choices. Thirdly, the discovery strategy is less ambitious in its concept of lead. In the iterative model, a lead was a temporary term for the most favoured compound of the day in an unrestricted and open-ended search for a superlative molecule as the development candidate. In the linear model, the lead is the molecule from the screening library best able to satisfy minimal preset lead criteria. The subsequent exploration of the lead chemotype for a congener meeting the requirements of a development candidate are unlikely to provide a better molecule than an exploratory search of more chemotypes. Fourth, the rigour and scope of the assay cascade is another casualty of the Rumsfeldian principle. The results of each assay in the cascade alone determine the composition of the much smaller set of compounds the next assay will receive. Thus, a molecule showing modest but adequate potency, which might achieve an excellent profile in subsequent assays, can be rejected in favour of passing through a more potent molecule with poorer prospects. In addition, the fast, facile assays suitable for the automated liquid handling needed to achieve timely processing of large numbers of molecules restricts the type of targets that can be assayed. Generally, assay development for novel targets is more challenging than for existing assay families. And fifth, a side effect was division of research into silos. At worst, biology and chemistry efforts comprised large independent teams without common goals and discourse. With 25 years of hindsight, there is enough time separation for a fair comparison of the virtues and pitfalls of the two paradigms to be made. Most notably, the cost defrayable against each launched drug has risen from considerably less than $1 billion to more than $3 billion. But the rate of output of new drugs has not increased. The proportion of late-stage failures has increased, and drug gestation time remains around 12 years. In the opinion of regulators, the novelty of small molecule output has decreased, and there has been little impact on important and urgent social targets such as dementia and novel antibiotics. The industry itself has not fared well during this era, having moved from a period of great growth with individual companies making breakthrough drugs that enjoyed many years of market exclusivity, to those same companies turning out poorly differentiated products, trying to compete in shared markets, and undergoing consolidation and downsizing. Many, even within the industry, have seen pharma's dulled performance as a reflection of the change in its discovery strategy rather than a coincidence, as evidenced by pharma's new practices of seeking novelty and innovation through open innovation and pre-competitive collaborations. Recent approaches to improve return. While the hardware and software of high-throughput automation and the scope and facility of secondary assays have grown steadily, the focus of effort in chemistry has been trying to fix shortfalls in the chemical library, serving the linear paradigm rather than instituting any alternative strategy, not least because of the large investment made in the overall linear strategy. Thus, the incompatible objectives of creating bigger and better libraries remain in place, and are only ameliorated by efforts to improve chemical library diversity through cheminformatics, or increasing the number of molecules displayed for screening by the implementation of improved automation for high-throughput chemistry and more efficient workflows. Chemistry, the numbers game. Success in opportunistic HTS relies on the timely screening of each of a succession of biological targets. Even with fast assays and good library curation, the economically optimal limit seems to be in the order of 1 million, notionally, drug-like molecules. Approaches to enrich the diversity of these screening libraries to improve hit rates with novel targets have been based on multiple regression analysis to score the relative contribution to biological activity of substituents in fixed scaffolds, or the physiochemical character of their substituents, or global descriptors of hydrophobic, electronic, steric, and other effects, through to the energetics-based plethora of descriptors of ligand deficiency used and argued about today. The concept behind all of these descriptors, however, is that existing molecules can inform the design of future molecules. While it is true that these tools can find better molecules within known drug space, again, it is the Rumsfeldian principle that prevents the finding and filling of holes in unfound drug space, where the unknown drugs for unknown targets might lie. These can only be found by active exploration of new chemical space, not passive screening or the synthesis of molecules claimed to be diverse, based only on an index that references similarity to historical molecules. Fragments. A Trojan horse? As an example of recent discovery approaches, fragment-based lead discovery, FBLD, recognizes the paradox that exists in HTS screening libraries, in that molecules included on the basis of their drug-like selectivity and lack of toxicity at historical drug targets are actually less likely to bind to unrelated novel targets. To overcome this, FBLD seeks to find from a library of fragments a low molecular weight fragment, or combination of fragments, that bind the novel target with only marginal affinity. These can then be intelligently elaborated into larger RO5 molecule leads with high affinity and selectivity for the target, using an iterative method. A key difference is the modus operandi in determining the affinity of the seed fragment and early derivatives, as their binding is too weak and the concentrations required too high to permit the use of routine biological assays. The perceived advantages of FBLD are that the necessary hydrophilic character, for solubility during screening, also ensures high ligand efficiency due to the high enthalipic binding in the elaborated product, which can be further enhanced by the entropic contributions of hydrophobic character without rendering the final compound too hydrophobic for use. In addition, combination of multiple fragments magnifies the screening power of the library by the square or cube relative to the same number of molecules in an RO5 set. The practical downside of the method is the complex biophysical technology needed to detect the marginal affinity of fragments. In this respect, the 2D protein detected NMR, used in the original SAR by NMR, work at Abbott to measure the changes in specific amino acids of the target disturbed by the fragment or fragments, has now been joined by a host of no less simple screening techniques. The need to obtain structural models of the protein fragment complex by X-ray crystallography is a further incumbent. With just a few reported successes and little knowledge of the failure rate, it is probably too soon to say how versatile and reliable FBLD can be as a tool for lead discovery. However, although there is some likelihood that the fragment libraries and targets of competitors overlap, it should not be a concern, as the fragment loses its identity in the very first step of a stochastic iterative elaboration to a full lead or even a product. In effect, it may be said that FBLD is just another way of seeding an iterative process. As iterative processes are stochastic, i.e., where they end up is highly dependent on what is measured and how it is used in the molecule design, it is probable that in different hands the same fragment or protein combination will deliver distinctive solutions reflecting the quality of the assessment and design steps. In fact, the same fragment could seed solutions for other targets in different therapeutic areas. However, in all cases, the versatility and the quality of the found asset derives at least as much from the iterative process as much as from the seed, including a propensity for obliquity when a discovery team is allowed to exercise their unfettered skills in pursuit of a superlative solution from even a hint of activity. Thank you for listening to part 1 of a strategic vision for 21st century drug discovery. You'll be able to listen to the rest of this article, as well as learn more about the authors, in the next episode. If you've enjoyed this episode and want more from Drug Discovery World, then you can subscribe to the journal and get the latest issues delivered to you via post or electronically, completely free of charge. Just head to ddw-online.com and you can subscribe there, as well as read further articles and view original PDFs of past articles, which have figures and images included, in our PDF archive. If you've enjoyed the podcast and found it useful, then please do subscribe and leave us a review. We'd love to get feedback. You can also follow us on LinkedIn and Twitter. Thanks for listening, and we hope to see you in our next episode.